So thanks for the opportunity to speak and for the invitation for this really nice drug-based symposium. And I actually am very proud to be the first speaker, so I can talk about target trends today. So really, I want to focus more on how we can track target innovation trends in drug discovery. Do you still see my screen? No, right? Because now it disappeared for me. Do you see it still? Okay. So I want to really focus more on target innovation trends in drug discovery. So really, how can we make use of this knowledge that is out there in the open domain to take more informed decisions in a drug discovery project? And I have to emphasize that the work I will present is mainly focusing on a paper we published last year. So this work I did in the framework of my role at the University of Vienna as a group leader. But since January, I'm also working for EBI, for the Open Targets project. And of course, there is also a nice angle to this work as I work now for Open Targets as well. So also I wanted to start with a little analogy to a science fiction movie I recently saw. It's not a new movie, so it came out in 2014 already. But I'm quite a late bloomer in science fiction, I would say, and maybe that's also a side effect of working from home. So, but anyhow, in this movie, which is called Interstellar, the main character actually moves through space and time in the search for a new planet, yeah? So he moves to the interstellar space, and as he moves through space, as I said, he also moves through time, and in the end of the movie, he ends up behind the bookshelf of his daughter's room, who now has grown up. So for her, time moved in a different pace. And I found this a very nice analogy, because as we think about drug space, we have to consider space, but I think we also should take into consideration time aspects, because all this information is readily available in the open domain. And since I'm the first speaker, I can also use this opportunity now to introduce you into some of the concepts. So as already mentioned just 5 minutes ago, we have to consider chemical space, but we also have to consider target space here. So as we look into chemical space, we all know drug-like space is vast. So people speak about the order of magnitude of 10 to the power of 60 compounds. Of course, this is a bit defined by certain rules, like basically you come up with this number if you define it by just 30 atoms and only 4 rings, etc., etc. But of course, if we consider the whole organic chemistry space, that's much larger. And in between these two, there is somehow the synthesizable space. Of course, we can only synthesize where we have reactions for. And then known drug space is a bit smaller. So that's basically what the known drugs populate. And this space somehow helps us to also define and set the boundaries, right, in our drug discovery campaigns. And the picture that you can see on the left-hand side, this is a very nice visualization that I took from the ChemMaps project. And the authors basically of this project, they also state that you can also use this even for making, for instance, toxicity predictions, right, for read across. Because you might imagine that you can look at a chemical compound which is close to the compound where you know the toxicity. So you could extrapolate from this neighboring compound. I think we have to be a bit careful there, because this is, of course, a multidimensional space that is compressed into 3 dimensions here. And we also know, right, that compounds that can have quite a different chemical structure still can quite have the same phys-chem or very similar phys-chem properties. So I think what we really have to do is we also have to take targets into account. So really these compounds, they bind towards certain targets, right? And they are mostly not very specific. So often we have promiscuous targets. So it's really interesting to study target space here. And as we talked about the chemistry space, which is vast, also protein space is vast. So I found differing numbers here for the human proteome. It's between 80 and 400K proteins large, approximately, but we are more interested, right, in drug targets. So the drug target space is nowadays, we say, at least 3,000 different drug targets. And people tried to really categorize these targets. So one initiative is this IDG initiative, so the Illuminating the Druggable Genome project, where actually targets were categorized into T-Clin, Chem, Bio, and Dark according to their target developmental level. So that would mean T-Clin is a target where there is at least one known drug. For that target, T-Chem means there is at least one chemical in Campbell, for instance. And for T-Bio would mean there is at least one disease annotated through OMIM. And the rest are the dark targets. So these are really targets that are understudied. And this project even defines those targets on their website. So I looked it up 2 days ago, and there is really a list of 144 kinases, a couple of GPCRs and ion channels that they are focusing now mainly. So I think that's a very nice initiative. In Europe, we have a similar project, Open Targets, and I just mentioned I'm also now working for this project. So I wanted to take the opportunity to shortly mention this. So in this project really, which is a public-private partnership as well, and multi-year project. So it started in 2014 already, actually. And there really we use human genetics and genomics data to systematically identify and prioritize targets. And I think this project is really also known for its target disease association score. So there are multiple evidence sources collected to date. It's 20 different evidence sources. And then this is summed up into an overall score. And what is also new now is really that there is an effort to try to include also target prioritization. So by including something like tractability and also safety. So that's this project, and I think it's really important that we have several projects in that domain. But as I said in the beginning, so I really, in my work, want to focus more on the time aspect, because in the medicinal chemistry literature, there is a lot of knowledge that we can make use of, right? So we can learn from the experiences that others made. And this work we started in 2016 approximately, first focusing on the chemistry space. So what we did is we actually extracted data from the Campbell database over 17 years. And from the compounds, we extracted scaffolds. And then we looked at scaffold-associated properties over time and wanted to really use those to characterize attention that the community pays to a certain scaffold, right? So what we wanted to characterize is something like scaffold popularity and what are good measures to look at scaffold popularity. And the measures we looked at were like new compounds per year, right, that have been published, number of bioassays, because all this tells you something about research attention. And another interesting thing we looked at was impact factors over time. So really, per year, we would calculate the median impact factor that the compounds in this year of a certain scaffold have been published in. And why did we even come up with this idea? Because this all was initiated by a blog post we read where people talked about biscalcones, which are covalent modifiers, actually, so paints compounds, and still people are publishing those compounds. And even we saw that they have been published in journals with increasing impact factor over time, as you can see on the right-hand side. So we wanted to see, can we actually reproduce this, and can we see this as a similar trend for other compounds? And in fact, it turned out in our study that chalcones, so these alpha-beta unsaturated ketones, are one of the most popular scaffolds nowadays still, or there is even an increasing interest in those compounds still. So that's how it all started, really looking at the chemical space. And also in this first paper, we asked ourselves, why is a scaffold popular, right? And we tried to explain a few things and trying to come up with an explanation, but we couldn't really conclusively explain this because I think there are many soft factors. So one thing we really computed was, we thought maybe synthetic feasibility of a scaffold might be a factor. So we computed the correlation between the slope of one of the measures we looked at versus the synthetic feasibility score, but we didn't see any correlation. So we don't think it's really driven by synthetic feasibility, at least not on the scaffold level, maybe on the compound level, but not on the scaffold level. But then there are other factors, right? And those are harder to capture. And those are things like intuition. Medicinal chemists, of course, who do organic synthesis since a long time, they're often driven by intuition and experience, and we have heard about this a lot in the literature even, and this is really hard to simulate, I think, even with very fancy methods like AI. There is also something like the Matthew effect, right? That's basically this effect that poorer people get poorer, richer get rich, and yeah, like in this context, it would mean we tend to use methods or even synthesis, methods that tend to work in the past. So there are so many factors, and it's really hard to really explain this conclusively. But now moving from this chemistry level to the target level, because here I want to focus more on time aspects of targets. We have to ask the question, what actually enables a target to be a good target for drug discovery? And there are also many factors that drive this, right? And quite obvious factors are things like the quality of a target, right? So really, what is the level of confidence that the modulation of a target will translate to a disease-modifying effect, right? That's target quality. Another thing might be tractability, or also called ligandability. So the likelihood of identifying modulators that interact effectively with the target. And even more desired would be a good druggability. That means the ability of a protein to bind a drug-like modulator, but then also with a therapeutically useful affinity, efficacy, and safety. And of course, we always have to keep the balance between efficacy and safety. So safety considerations have to be also taken more into account in terms of more systematically on the target level in the future, I think. But apart from these factors that we all know about, and you can read about them in literature, and I've mentioned here a very nice literature source, I think, are other factors. And I found this nice table that you can see here in the right corner, which talks about the commercial impact of drugs according to target classes. So it's kind of interesting what the market share is of certain target classes, and also how many active pharmaceutical ingredients are there per target class, and then also setting this in relation to targets. So for instance, what you can see is, of course, GPCRs. So this is really the state of the art 2015. It might have changed a bit, but I found this on a very nice poster in Nature Reviews Drug Discovery. And I haven't found a newer one, but still it shows a trend. So GPCRs, okay, there are many targets used from this big class of protein targets, but also a large number of APIs. But then there are others like kinases. Okay, now to date, there are a bit more than 50 drugs approved on the market, which belong to the family of kinases, and not as many targets yet as GPCRs, right? And then what you can see as well is ion channels. There are not so many targets actually used from the family of ion channels, but there are a lot of drugs actually that target ion channels. And then also interesting, the commercial impact of these. So this might also drive our decision, right, on which target we want to focus on. And then there are other factors that I haven't even listed here, which I read about funding, for instance. So funding also seems to be biased. So there seem to be more grants going out for certain target families and not so much for others. So we wanted also to look into this a bit more on a higher level. So really looking at over time the popularity of target families. And we thought about what could be good measures to characterize attention here. And again, like in a bit similar to the previous paper, we looked at new compounds, not new compounds per year, but compounds published per year in Campbell for each target family. So the different colors encode the different target families, and also unique targets per year, and papers, that's the right upper corner, PubMed IDs, and then also we looked at drug annotations. And what you can basically see is that GPCRs, so that's the green line, tend to have a high number of compounds and papers over the last 10 to 15 years, basically. But the last few years seem to be a bit like this trend has changed a little bit now. So kinases seem to have outpaced GPCRs in recent years. And what is also interesting is that how like actually the shape of the curve, so to say. So for kinases, if you look into targets, into the curve for targets, you really see there are peaks. So this is due to a few studies only, because you can also see the number of papers is quite low for kinases in comparison to GPCRs. So for kinases, there have been these large kinase selectivity screens. So a bunch of new targets has been published in just 1 or 2 papers. So that's also interesting to know. And for GPCRs, actually only, so 33% of all small molecule drugs are targeting GPCRs. But only 12% of human protein targets. So that's also quite interesting. And then if you look at this drug annotation curve here, so that's basically capturing drug efficacy target annotations as they are captured in Campbell. And this peak here at the very end in 2017, I looked it up today because I thought maybe that was a mistake, but actually it's really just one paper reporting these many drug efficacy annotations. So that's not unique drugs, that's really annotations as they appear in papers, and even if the same drug is reported multiple times in a year. But probably this means that is not a very good measure to capture attention for a target family. Sorry, now okay. So that's the next slide. So, but then we asked the question, okay, now knowing on this high level about target families, what drives the attention basically, how can we decide into which targets to invest? And one thing we can do is we can actually annotate targets with these target developmental levels that IDG developed. And we did that, and we found out that over the year, the picture is a bit changing, right? So whereas for GPCRs and ion channels, you can see that this category T-CLIN, so that's really targets with at least one approved drugs, is really high and not really changing a lot. But for others, like for instance, nuclear receptors, that's really in percentage. So really it shows this in relative numbers. So we really see in the last few years, nuclear receptors have really, there were a few new targets that don't have a drug, where actually just a chemical is reported. So this might offer new opportunities actually for drug intervention. And next, we then also wanted to know about biological aspects of target innovation trends. So how can we do that, right? So basically, it's really looking into target innovation trends over the years, but then you can actually annotate targets to gene ontology, biological process annotations, and on the other hand, you can annotate them with diseases, and we took them from DisGenet. And then you basically get a disease trend and a biological process trend line. And we did this actually per target family, because we wanted to really see the contribution of the different target classes to this trend. You can, of course, also do this across all targets, but then trends would potentially look a bit different. And then a trend who probably for one target family looks like a steep positive trend could across all target families look like a negative trend. And I will show you this on the basis of a few examples. But I really have to emphasize here, we don't know if there is a causality. So what we are doing is really looking at interest in targets over the years as it is captured by Campbell. But we don't know if these papers have really been published because people were interested in this exact disease, right? But it still shows something, because we know there is an association of this disease and with this target or target family. Okay. So let me give you an example. So this is showing trends of biological process annotations for kinases. And one steep increasing trend was the trend for immune system processes. So we really saw that targets related to immune system processes in the target family of kinases are receiving more and more attention over time. And then looking into the contribution of the different protein classes, we saw that the family of Janus kinases here contributed a lot. And then going into literature, I basically found out that indeed the last 2 years there have been two drugs approved that are Janus kinase inhibitors for the treatment of autoimmune diseases. So this especially for rheumatoid arthritis. And so these might be really interesting target classes to focus on. And Janus kinases are non-receptor tyrosine kinases, so they are essential mediators for downstream signaling. So really, really important for the immune system. So it really makes sense what we see. And on the other hand, looking into the protein class of GPCRs, we saw a downward trend of circulatory system processes, which was a bit astonishing for us to see that. And this also basically is attributed to less interest into the family of dopamine receptors, adrenergic receptors, and adenosine receptors. So these protein classes are not so much investigated anymore like in past years. And basically, we couldn't believe that. And then again, I looked a bit into literature and found out these days now GPCRs aren't so much in the focus anymore for treating cardiovascular diseases, or at least for coming up with new leads. It's more really now downstream targets like beta-arrestines, for instance, where the focus of research is. And now moving from biological process annotations to the level of diseases. For some of you, this might even make a bit more sense to look at those innovation trends at the level of diseases, because this really explains to which extent can the involvement of a protein in a certain biological process be translated into the development of a pharmaceutical for the treatment of a particular disease. And again, we did this analysis on the protein family level. And what you can see in this table that I'm showing is that for kinases, we basically saw only positive trends. And interestingly, most of these diseases were related to cancer, right? You see different forms of cancer here, but also intellectual disabilities, schizophrenia, for instance. But for all the other protein families, we exclusively saw negative trends. I have to say, of course, we found much more trends that I can depict here, but this is basically a list of statistically significant trends. So we did some previous filtering, of course. And on the right-hand side, you can see an example for instance, the trend for malignant neoplasm of prostate, where also Janus kinases seem to play a role in this increasing interest. And on the other hand, for GPCRs, bradycardia was one of the diseases where the trend was going down. But again, we don't know if that's really the case in terms of causality. So really here, the interest in the targets that are annotated with this disease has gone down. But this also nicely fits to what we have seen previously for the decreasing trend in cardiovascular diseases. And so presenting this work at several occasions and talking to other researchers, we often got the question, how can you make use of such a tool, right, in drug discovery? And can you maybe use it for forecasting, right, for making predictions? I think we're not yet at the stage of predicting anything, but what we can nicely do is there is a web app that we developed as part of this paper that we published last year, where you can actually explore target trends, disease trends, and gene ontology trends. And I just made a few screenshots because usually a live demo doesn't work so well. So you can see, for instance, for the biological for the disease intellectual disability, you can then also look at the contribution of the different target families towards this trend. And on the right-hand side, for instance, you see some trends for targets. You can also overlay these trends. So this becomes quite handy if you're interested in a particular disease or target. You can really nicely explore what has been done, at least to the level of what is stored in the Campbell database. And I think such an analysis or such a tool will even make more sense in the future as we include more data sources. And one source we might think of is clinical data, because Campbell really captures preclinical data. So what about including clinical data? We tried this out and included this in the paper as well. So what we did here is we had 237 significant disease trends in our study. And what we did is, for those diseases, we also queried clinical trials. And then for those diseases, of course, looking for several synonyms of the disease, and then we just overlaid the trends. And of course, we have to appreciate these are very different time points that we are looking at. But if you look at the left-hand plot for AML, you see that the preclinical trend, so the trend for the target trend from Campbell, is steeply increasing. But if you look at the trend from clinical trials, you see it's rather flat. So this trend hasn't been taken up by the clinics, I would say. And for schizophrenia, we see a downward trend both for clinical studies as well as it's captured by papers in Campbell. So I think there can be much more done in the future by including more data sources, and also think about how do we accommodate these different data sources in terms of the time lag and so on. And one thing I also mentioned is, okay, to be sure about causality, we probably have to include text mining approaches to really, from the paper, capture the disease that the people have been working on at the same time. And finally, we also were wondering a bit about causality in this study. So as I mentioned a few times, we can't guarantee causality, but we wanted to systematically explore this. And what we did is we actually also queried PubMed independently of Campbell, really looking into the paper titles and abstracts and looking for the disease terms that we captured, and then counting the papers for these diseases over time, and then really overlaying the plots for the target trends from Campbell. And we only found 18 Granger causal disease trends in our study out of 2,000. So that's not really, we are not very happy about this, but still, I think there are a few limitations here, because on one hand, PubChem and Campbell, of course, cover not exactly the same chemical space. On the other hand, maybe the counting papers might not be the best way to capture attention, right? So we couldn't capture here bioactivities, for instance, which would maybe better characterize attention. But on the other hand, when looking at correlation only, we actually saw that 22% of all trends were correlated. And we thought maybe that's just a random finding, and that's why then we shuffled the data. So on one hand, we shuffled the values, on the other hand, we shuffled diseases, and in both cases, really the correlation was much lower or poorer. So we think there is something in the data. Maybe it needs a bit more testing and including more data sources to make this work even better. But I think that's a very interesting research direction to go. And having such a tool can actually really help you guide decisions in drug discovery if you make use of it. So that's basically my last slide, my take-home messages. So I think time trend analysis can really help guide decision-making at a very early stage in drug discovery. And by including these target developmental levels, these categories that we have for different target families, it really reveals areas of interest in target space that have just emerged in literature maybe. So it really points you to target space that might be interesting to focus on in the future. So it highlights novel opportunities for drug intervention. In terms of causality, this needs to be explored a bit further, I think, and I think that's a necessary next step to see if target trends are really descriptive of disease trends. And finally, I think integrating more data from the clinics, maybe from patents as well, might offer an additional interesting source of information to even go beyond what we already did. So I want to thank all the people involved in this work. So co-authors from the paper, especially Rajarshi, who is really a very great discussion partner, contributes a lot of ideas. Also Lars from the University of Vienna, who also helped with the computations a lot, and Nathan, who also was an important discussion partner. I also want to thank Vishal from NCATS, who provided us with the mappings to the target developmental levels, and from EBI, Andrew and Ian, who just recently started to discuss these topics with me as well. So I'm hoping there will be some further developments in the future, and many thanks for listening. I'm happy to answer any questions now or later in Gather Town.